Hello and welcome. This is A&P Live Studio. I'm your host, and as a registered nurse and an anatomy and physiology instructor for advanced healthcare professionals, I'm here to guide you through some complex topics. Today, we're going to be talking about a really important neurological condition, multiple sclerosis. So, to really get a handle on what multiple sclerosis is all about, it's this chronic, immune-mediated disease of the central nervous system. I think the best way to start is always with a patient's story. It really helps to ground the science in a real-world experience. Let's meet Sarah. She's 32, a graphic designer, and what she's experiencing is actually a super common first encounter with MS. She's got blurry vision in her right eye, we call that unilateral optic neuritis, and this weird tingling, asleep feeling in her fingers, which is known as paresthesia. So, her symptoms, they're really our first clues, our way into understanding what's going on at a deeper pathophysiological level. Okay, so here's our plan. We're going to follow Sarah's story to see what's happening down at the cellular level. Then we'll connect that back to her symptoms, look at the very latest in how we diagnose and treat MS, and finally, we'll peek into the future of MS care. It's going to be a comprehensive look. All right, so before we can really appreciate what's going wrong in MS, we've got to take a quick step back and review how things are supposed to work. Let's talk about our nerve cells, and specifically this incredibly important stuff called myelin. You could say the myelin sheath is really the star of this whole story. The best analogy? It's like the plastic insulation around an electrical wire. It's this fatty layer that wraps around our nerve fibers, our axons, and it's absolutely crucial. It allows electrical signals to just zip along, super fast and efficiently, all through our central nervous system. I mean, this lightning-fast communication is what lets us do, well, everything, from seeing the world to moving our muscles. And right here, this image just lays it all out. It's the core problem of MS in a nutshell. On the left, you've got this healthy neuron. See how thick and complete that myelin sheath is? But then look to the right. That's a neuron from someone with MS. The myelin is all damaged. It's been stripped away. We call that process demyelination. And when the insulation is gone, what happens to the signal? Well, it gets delayed or twisted, or maybe it just stops altogether. That is the fundamental disconnect that leads to all the symptoms we see. So that leads us to the big question, right? What on earth is causing this damage? Well, it turns out the culprit is our very own immune system. It's basically a case of mistaken identity. You see, MS is an autoimmune disease, which is just a fancy way of saying it's friendly fire. It all starts out in the body, in the periphery, where for some reason T cells get activated incorrectly. A virus, maybe something like Epstein-Barr, might be the trigger. Then these rogue T cells figure out how to sneak past the blood-brain barrier, which is supposed to be this super selective security gate for our central nervous system. Once they're inside, they start sounding the alarm, releasing all these inflammatory signals. This calls in backup more immune cells like B cells and microglia, and this whole coordinated attack is what leads to the final devastating step, the demyelination, that's stripping of the myelin, and eventually even damage to the nerve fiber itself. Okay, let's bring this all back to Sarah. How does all this cellular chaos explain what she's actually feeling? Well, it's pretty simple, actually. The signs and symptoms of MS depend entirely on where in the central nervous system these attacks, these demyelinating lesions, are happening. It's all about location, location, location. So if a lesion forms on the optic nerve, boom, you get optic neuritis, just like Sarah's blurry vision. If the attack happens on a sensory pathway in the spinal cord, that can cause that tingling and numbness, the paresthesia. If it's in the motor pathways, the pyramidal tracts, you might see muscle weakness. And if the cerebellum gets hit, that can cause problems with coordination, what we call ataxia. You see? It's because the lesions can pop up all over the place, in the brain, the optic nerves, the spinal cord, that MS can cause such a huge range of symptoms. Now, from a clinical standpoint, it is so important to understand that MS isn't just about these inflammatory attacks. Over time, the disease can shift. In the beginning, you have these flare-ups and the body even tries to repair the myelin. But after repeated attacks, the damage can become permanent. The nerve axons themselves get injured and are lost for good. This can even cause the brain's outer layer, the cortex, to actually shrink. We call that cortical atrophy. And it's this permanent, irreversible neurodegeneration that really drives the long-term progressive disability we sometimes see in MS. Okay, so how do we actually put all the pieces together and confirm a diagnosis? For a long time now, the gold standard has been something called the McDonald's criteria. And what's really exciting is that they were just updated in 2024, making it possible to get an even earlier and more precise diagnosis than ever before. So let's just quickly walk through these updates because they're really important. First off, the optic nerve now officially counts as a location for lesions. This makes it easier to prove that the damage is happening in different places, what we call dissemination in space. Next, when we look at the cerebrospinal fluid, we're still looking for oligoclonal bands. Those are unique antibodies in the CNS. But now, a test called the kappa-free light chain index can be used too. It's another great marker of that B-cell activity we talked about. And finally, the new criteria really embrace advanced MRI techniques. Things like the central vein sign, which helps us be sure a lesion is from MS, and paramagnetic rim lesions, which tell us about chronic, smoldering inflammation. Why does all this matter? Because diagnosing MS earlier and with more confidence means we can start treatment faster, and that's our best shot at slowing this disease down. All right, so once we have a firm diagnosis, the mission becomes clear: manage the disease and slow it down as much as possible. And thankfully, the world of MS treatments has absolutely exploded in recent years. We have a whole arsenal now, what we call disease-modifying therapies, or DMTs. And they come in all shapes and sizes, from older injectable meds to a bunch of different pills and some really powerful infusions. The end goal for all of them is the same: to calm down the immune system and stop it from attacking the central nervous system. But they all go about it in slightly different ways. Some are more of a broad approach, while others are incredibly targeted, aiming to stop specific immune cells in their tracks before they can even get to the brain. Now, some of our most powerful tools are the anti-CD20 monoclonal antibodies. Honestly, these have been a total game changer. Their mechanism is so elegant. They're designed to find and take out a very specific type of B cell, the ones that have a protein called CD20 on their surface. By just removing this one key player from the game, we can dramatically interrupt that whole inflammatory process that causes MS relapses.
And the field isn't stopping there. The next big thing is figuring out how to fight the inflammation that's already inside the central nervous system. And that's where a new class of drugs called BTK inhibitors comes into play. These are really cool because they're small molecules. So unlike some of the bigger drugs, they can slip right through the blood-brain barrier. And this is huge. It means they can get inside and quiet down not only the B cells, but also those angry microglia right at the source. The hope is that this will tackle that chronic, low-level, smoldering inflammation that we think is a major driver of long-term progression.
So all of this naturally leads us to think about the bigger picture. Why did Sarah get MS in the first place? And what does the future hold? Well, it's not a simple answer. It really comes down to a complex dance between a person's genetics and their environment.
Just look at this number: 25%. If one identical twin has MS, the other twin has about a 1 in 4 chance of getting it too. Now compare that to the risk in the general population, which is way, way less than 1%. So that's pretty powerful evidence that genes play a big role, right? But it also tells you something else really important. If it was only genetics, that number would be 100%. So clearly, something else is going on. And we're starting to figure out what those something else's are. We know there's a specific gene variant, HLA-DRB1-1501, that carries the biggest genetic risk. But you need more than just a gene. You need an environmental trigger. It seems that a past infection with the Epstein-Barr virus is almost a requirement. And on top of that, things like low vitamin D levels or being a smoker can also push that risk even higher.
So here's the bottom line, where we are today. Our current treatments, they're fantastic at putting out the inflammatory fires that cause relapses. But the next big challenge is really a two-part mission. First, how do we protect the nerves that have already been damaged? And second, and this is the holy grail, how do we get the body to effectively rebuild that lost myelin? How do we promote remyelination? And that really is the ultimate question that drives all the research now. We've gotten so good at controlling the immune system part of the disease. The next giant leap forward has to be a shift in strategy. We need to move from just playing defense to playing offense, to find ways to actively repair the nervous system and maybe even restore the function that's been lost. Stay patho-curious, and I'll see you in the next AMP Live Studio. Please remember, this video is for educational purposes only and is not intended as medical advice, diagnosis, or a substitute for professional healthcare decisions.